A couple quick things before we do get started. The first one is that if anybody wants to take a totally cool lab course and have time Monday and Wednesday afternoons, the recombinant DNA lab, right, Damon? It's a really cool lab. He took it last year. Basically, you work with viruses, kind of like what we're going to be talking about today. Currently, it's really small. I think there's something like 6 signed up for one section and 7 for the other, or 8 and 7, which means it should be really great. I've got some wonderful graduate students, namely my graduate students, who will be teaching that class. Should be a lot of fun. If you're interested, I think you might have some time. Again, there are 2 sections, Monday and Wednesday afternoons from noon to 3 and 3 to 6. So those are the times. But if you're interested, come and talk to me after class today. There's also, and I'm sure there are probably some of you who are doing this as well, tomorrow morning, the crack of 8 a.m. Is the molecular genetics course, which I'll be holding over in the Urban Center building. It was completely full when people started to try and sign up, and I guess I scared a whole bunch of them away. And so there are now some spots in that class. If you're interested in taking that again, come talk to me, and we can talk about a little bit about that later. But of course, this is the most important class, the coolest subject ever, virology. And so we're going to be talking about just a little bit, again, about virology for the next 10 weeks or so. I'm actually going to talk about every single one of the viruses that's listed up here on this slide. Talk a little bit about cowpea chlorotic mosaic virus, CCMV, up the top on the, I guess, your left, my right here. STIV, which is a virus I discovered in the middle there at the top. SSV, which is what most of my lab actually works on, up in the other corner there. Talk a little bit about adenovirus down here in the middle. And then one of my favorite viruses outside of the viruses of Archaea, bacteriophage T4. Start out just basically with some stuff about the course. How do you find me? I'm over in 466 SRTC, the building previously known as SB2, for those of you who've been here for a while. Office hours are right after class. How many people have something right after this class? Bunch of you. Okay, so by appointment is certainly an option as well. I'm here basically from about 8 to 5-ish most days of the week, at least Monday through Friday. More than happy to talk to you there. Also, I'm email dependent, so send me email. I usually get back to people really pretty quickly on that, unless I'm completely swamped. There's D2L mail, which I maybe get to once a day if you're lucky. So basically my PDX email. If you really want to call and talk to my voicemail, go ahead. That's up to you. Prerequisites. If you have not taken molecular biology, how many of those of you? I don't know your names, so that's okay. This course will be a challenge because I'm going to assume that you know the material that was in molecular biology. And so that being said, I've had people who haven't taken molecular biology who've gotten A's in this class, and I have people who haven't taken molecular biology who have not gotten A's in this class. So again, I'll just assume that. I'll assume cell biology, but a lot of the cell biology, and I know I've talked to a number of you about this already, it's mostly having to do with cell structure, and it's not that critical, and particularly if people are taking cell biology at the same time as this class, that's fine. And again, if you've got some more questions about that, come talk to me again right after class or office hours or email, you name it. My apologies to people who want to learn about how viruses make you sick. This is not the right class for that, and there are 2 reasons for that. One is it's mostly has to do with immunology, and number 2, I know very little about it, so trying to teach you would be a bit of a challenge. Plus, there's nowhere near enough time to cover the molecular virology, which is what I'm really interested in, as opposed to pathogenesis in 10 weeks. So we're just basically going to talk about the main classes of viruses. A lot of people, when they have a virology course, will often set it up in one of 2 different ways. One is to go through the various different families of viruses, like we're going to, and another one is to concentrate on mechanisms. And they're really sort of 2 ways of looking at exactly the same thing. Really, how amazing viruses are in terms of what they do. And you can either take that from the examples of the individual viruses and how they do it, or go through how they do it relative to each of the individual viruses. I find it easier to remember if we talk about the different virus families separately, and then if any of you are moving on into a more medical career, that's how people talk about viruses. You don't talk about the replication of viruses. You talk about filoviruses or Ebola. Anybody reading about Ebola in the news recently, or Ebola, depending on your point of view? There's an outbreak in Guinea, and we'll talk much more about Ebola later. But basically, almost every day in the paper, on about page 5 or something like that, there's something about that. I guess now, how many people actually read the newspaper these days, other than me? Okay, I guess it's just me. But there's stuff online about it as well. So basically, DNA, RNA, and retroviruses, which actually have a lot to do with how these things replicate in the first place. Bacteriophage to HIV-1, we'll talk about some other viruses as well, but sort of simple to complex. But the real sort of major thing here really is about virus replication. Yes, we're going to talk about the individual families, but the main focus is here is how those viruses are making more viruses. So that has to do with genome replication, how the virus makes more of its own genome, how it expresses the genes that it has in its genome, and how viruses put together, and I should really say the virions, the extracellular part of a virus here. These are really specific to viruses, but they use cellular machinery. And so viruses are actually a great way to understand how cells work as well as how viruses work. And again, I'm not going to talk very much about pathogenesis at all. Unfortunately, the class, we have to go through this process. What do? My least favorite thing to do, probably your least favorite thing as well. Midterms, there are going to be 2 of them, and a final. Each of them is worth 30%. First midterms on the 21st of April, second one's the 14th of May. There'll be a non-cumulative final on Tuesday. It's the regular exam time for this class, also at 8 a.m. All the exams are 50 multiple choice Scantrons. Again, that has to do with the number of you and the number of people helping me grade this class, which is this many. There are lots of old exams and examples on D2L. One thing to bear in mind, however, about this is that those old exams were based, at least some of them, on just having 1 midterm instead of 2 midterms. I used to have a problem set, but then it would take me probably about the whole amount of time between the problem set and the end of class to actually grade it. So we're just doing midterms now. So some of the exams, for instance, there'll be one exam which will have half the class on it. It's now been split up into 3. Clickers. Basically, clickers, yes, I've got my cute little instructor one here. The main point about clickers is really for me to try and get a handle on where you stand. And so it's as much about information for me as it is torturing you with clicker questions. And I try and use that. I'll be having daily quizzes. They'll be starting in week 2. So this week you don't have to worry about them. They will be worth 10% of your grade, and the only reason for that is to make sure that you actually come and use them, because if you don't use them, the whole feedback point of view is kind of gone here. So that being said, anybody have clickers with them? Yeah, question. Okay, good, great. So please bring them out and answer this question here. Again, it's not going to be graded. This is, again, more for my information than anybody else's. You're a junior biology major, senior biology major, post-bac, junior, senior non-biology major, or everyone's favorite, none of the above. And I happen to know at least one of you is none of the above, although he may not have his clicker with him today. So only one person has voted. So there's only one of you who's going to admit to this?
It won't let me vote.
Yeah. Status blinky blink. They're not working here. 2 people have managed. I'm not quite sure how that works yet. They're all doing blinky blinks.
Do we have to re-register them for the
Well, it should be. I think it probably has to do with what channel they're on. You will have to re-register from this term, but I think if you screen through channels, it should, this says AA here, so hopefully that would be. Again, it's not that critical that I have information on this. The main reason, actually, for asking this question is when I go through, and I'll talk about normalizing grades in a second, so that I actually try and normalize grades to a class that actually fits with you, so that all these annoying post-bac students here don't clearly mess up the curve for everybody else. Or maybe it's actually the junior biology majors who are screwing up the curve. You know. But again, just so that I have an idea here. Okay, so there are 10 of you so far. Okay, so registering clickers would be a good thing. Yeah?
Is there a code we have to enter to?
No, it should be. So it should just be, yeah, channel AA should be what works.
It's on the channel, but it's just the numbers. It's only
Exactly.
Oh, okay. Well, maybe it's set on okay. I'll recheck it for next time, so we'll have another one which you can check. This is also a reason why I do week 2, so I can work out what all the problems are. Yeah.
Should we try to register them?
Pardon?
Should we try to?
Yeah. No, definitely register makes that's something completely independent of the numbering versus letters. So, which actually is okay. We can stop this. Register them. Yes. Let's see. Of course, now this is even talking to me anymore. Let's see. Click on here. Again, this is the whole technology issue here. So register online. Don't register in class. I found that takes far too long. And the online registration, at least in the past, has worked really well. And in fact, first time with the clickers usually worked well here too. So April 18th, which is right before the midterm, name, clicker number, and your Odin ID. Grading, again, why I wanted to get that data, and maybe I'll just ask exactly the same question on Wednesday. That might be a good idea. I can do the same question on Wednesday, see how many more answers I get, see how many people have changed their answer between the 2 times. Grading is going to be normalized to whatever the high score is in the class. And that will be whatever high score is that is not some graduate student or something like that. So top score will be set to 100% and then just scale from there. This is exactly what I've done. How many people had me for molecular last term? Okay. So it's basically exactly the same thing there. My exam policy, closed book, no time limit. I think there's a class in here right after this, but we can always move outside in the hallway and finish up. I never liked having time pressure on my exams, but that being said, as soon as somebody's left, then you can't start the exam because I let people take the exams with them. So just be here on time on exam days. This should be pretty straightforward, cheating, 0, etc. Attendance is not required. I know life happens. I'm one of those people as well, although, once, actually, this was a couple of years ago, I actually was in the ER the first day of class one term. But highly recommend coming, and the main reason for that is just looking at how people do with their grades. Self-reported, people who always came did much better than people who came rarely. This is, again, before I had those 10% on the clicker questions. I also understand, some people may have something else they really need to do soon thereafter. I'm always going to put clicker questions right at the beginning of class. And after about the first 5-10 minutes, if there's something you really have to do, some exam you really have to study for, just please sit on the sides and leave quietly when you're done. I totally understand that. Makeup exams, again, life happens. Hospitalization, death or birth in your family. I need written evidence that's mostly to protect my posterior. And that exam will be essay, and you can talk to people who've had my essay exams before. I recommend being here. The regrades must be requested in a week, and I may or may not get around to scanning your Scantrons. And again, if you mess with your Scantron, so be it. How do you do well in this class? Highly recommend the textbook. I've gone through, I think this is about my 4th textbook now. I really like this one for a number of different reasons. One, it's about molecular virology. But it has a couple of things in it that I really like. The chapter overviews, each of the individual chapters has a really nice overview at the beginning on basically the important stuff in that particular chapter. And particularly interesting for the comparison, because I really do a lot of comparisons between different viruses here. There's a nice glossary, and they're pretty good references. I post, anybody get my lecture notes already? Yep, so I try and post them by 10 o'clock the night before. One of the things that, again, people who were in my molecular biology last term will know is I record my lectures, and in fact I recorded my lectures last year for virology. So last year's lecture recordings for most of the classes are going to be posted. I'm not sure if I can put them all on D2L because they're pretty big, but I'll try and do that, at least have them there, or maybe on a Google Drive. Other thing, of course, is to ask lots and lots of questions, because it, A, slows me down, and B, hopefully is going to, if you have a question, somebody else probably didn't completely understand that either. D2L has the syllabus, lecture presentations, old exams, supplemental materials. So there are going to be a couple of things that are not in the textbook, particularly bacteriophage T4. There's also some work on some new viruses that my lab discovered, which I'll put on there. Discussion forum, this is mostly for you. I will check it every once in a while, but it's really useful, I think, for you. And then mail, but as I mentioned before, I don't really read that often. There are more or less infinite numbers of resources as far as virology is concerned. Principles of Virology, which is the textbook I used to use, and you'll see a number of figures that I use from that textbook in the lecture here. I'll have at least one copy on reserve in the library. It's not there yet. I need to do that today. It's a nice reference text for animal viruses, but it really pretty much only covers animal viruses, and that's one of the big problems that I have with it. I think that constrains things a little bit too much. Again, second edition will be on reserve there. I don't have the third edition yet. Fields Virology. Fields Virology is sort of the holy book of virologists. I think it's 3 volumes now. It's about this thick. It's an amazing reference. There's a lot of stuff on Google Books. In fact, most of it's on Google Books, so you can access it there. I think it's on the 5th edition or something like that now, but the previous ones are pretty good. It's a very active field, so Journal of Virology, Virology Science and Nature, all of these things are accessible through the PSU library, so you can get access to these things. I have my own virus library that you can borrow things from on pain of whatever grade it is you end up with a class. That's in my office, and so office hours as far as class is concerned. There's also a ton of stuff on the web, and in fact, one of my favorite sources for virology information, and I think actually a great resource for you guys, are Vincent Racaniello's virology lectures. Any of you know or have heard of Vince Racaniello? TWIV is one of the things that he does, which is This Week in Virology. It's usually a lot of fun. It's kind of long, gets a little bit long-winded, but other than that, generally extremely good, particularly some of the earlier lectures. He has a Coursera course and his virology class that he teaches at Columbia. I just listened to his introductory lecture the other day. In fact, I'm using some of that in my lecture as well. So I highly recommend, if you want any more information, go and take a look at this. It's a little bit different approach to some kinds of things about virology. He teaches, however, in the different mechanisms versus different viruses, so things sort of go apart after the first couple of lectures. There's some really great virus structures out there, virus taxonomy, all kinds of information on the various different families. Again, I have on here buyer beware. Some of the stuff on there is not completely right, but at least it's a good start. This is actually really pretty similar to what I was talking about, the overview, each of the chapters in our textbook. A lot of that's there. The Virus Focus Group, which is a website that I run about astrovirology, which sort of a term I was named an astrovirologist at a meeting I went to. It's like, really? Is that what I do? Okay. So, but we're going to try and post some stuff on here as well. Special stuff about this class, bacteriophage T4. I'm willing to bet with our textbook, I didn't bring my copy with me, I'm sure the author of the textbook asked somebody to write a chapter on bacteriophage T4, but they probably didn't get around to it. I can't explain any other way why it's not being covered, because it's an absolutely critical aspect of these things. We'll have a couple of visiting lecturers, or maybe the visiting lecturers are going to be a DVD. We shall see. So I'll be at a meeting in the sort of third week of May. Ryan Estep unfortunately emailed me last night and said he can't come and talk about herpes viruses, and then the pox virus lecture as well. I'll talk a little bit about some of the viruses that we work on in my lab, particularly this RNA-DNA hybrid virus, sulfolobus spindle-shaped virus, and sulfolobituridae icosahedral virus, which is the one that's up in the picture there. I'm working on a movie. We'll talk about this a little bit more later. The filmmaker keeps saying, oh, I'm going to come and film your lecture. It's going to be awesome. He's been talking about this for the last, 2.5 years, I think. So we shall see if it actually happens. I will be doing a science pub. You guys know about OMSI science pubs? At the Mission Theater on April 15th. Should be a lot of fun. And with any luck, you guys will be able to ace the trivia questions that they have at the beginning of them. So I'm also thinking about, since I've recorded all of my well, not all of them, as many as I could. We had some technical issues last year as well. Maybe flipping some lectures, asking you to listen to the lecture before you come to class, and then we just talk about it. So I'm going to try and do that. I haven't quite figured out which one is going to be the best to do that with. Unfortunately, virology is a really fast-moving field, and so some of the stuff that I will have talked about last year has actually changed since then. Any questions about the class at this point? Yeah.
So the questions at the beginning of class,
yeah,
those are last lecture that's being covered?
Yes, those will all be from the last lecture, unless I do a flipped lecture format, in which case they might be on that particularly flipped lecture.
Okay.
Yes? Dan?
Are we going to need to give numerical answers via clicker, or can we use the iClicker ones?
The iClicker ones are fine. Yeah, A, B, C, D, E if I get around to coming up with enough potential answers. The other thing, which I completely forgot to mention, is that, again, those of you in molecular know, but I'm recording these lectures. That's why I've got the wireless mic and all of that. I'm also getting screenshots of my computer here. All of that gets posted on YouTube after class. And so you all have access to it. Anybody has access to it. One of the things I'm going to do today is actually split this lecture into 2 things on YouTube, because not everybody needs to know about the details of the class at the beginning. And the other stuff is much more interesting. And that brings me to one more